Hello and welcome dear viewers to the video on dissolution testing frequently asked questions. In this video we are going to see which are the frequently asked questions and how these are answered. Dissolution is a topic of great interest for the interviewers, and if the professionals are knowing about the dissolution testing and if these professionals can answer the questions, then certainly it will have a great impact in the interviews. Let's start with the video. Now, whatever the questions I have selected, these are generally asked in the interviews on the basis of the frequency I have included. And whatever the answers I am going to give, these are not the only answers. You can search the answers and make your own answers. First question is why dissolution testing is done. So always the dissolution test is done to see whether the formulation is having suitable dissolution characteristics or as per the specified requirement for dissolution testing or not. Dissolution testing is a performance testing. The dissolution testing serve as a in vitro testing to predict the in vivo behavior of the formulation. That's why dissolution testing is done. Second question is what are the USP dissolution apparatus? So 7 apparatus are there, and out of these generally 4 apparatus are used for oral solid formulations. So apparatus 1, 2, 3 and 4 are mainly used. Out of these 4 apparatus 1 basket and paddle apparatus is most widely used for the oral formulations. Then what is QC media dissolution? So QC media dissolution or QC dissolution testing is the testing which is performed onto the approved formulations to release the batch in the market. In the quality control department, this dissolution testing is performed as per the specified parameters and it is checked whether the batch is complying to the predetermined specifications or not. Fourth question is what is discriminatory media or discriminatory dissolution? Discriminatory dissolution is that dissolution test and the parameters and the specifications which can distinguish between the batches which will be bioequivalent and which batches will be bioinequivalent. These methods for discrimination can reject the batches which may not prove to be bioequivalent. The discriminatory method of dissolution can detect the batches which are made or which are manufactured without the specified ranges of the process. That means, in simple words, the discriminatory media can detect the batches which are mismanufactured, like hardness of the tablet. If the tablet is too hard, then this method should detect the batch, and based on that discrimination, the batch is rejected. So QC media and discriminatory media is always asked. See, QC media should be discriminatory media, and nowadays regulatory authorities are asking for the QC media should be discriminatory media. What is biorelevant media? So generally the biorelevant dissolution is performed before the bioequivalence or bioavailability studies, so that the behavior of the formulation with the media composition similar to in vivo conditions can be done, and based on this biorelevant dissolution, the prediction can be made for the formulation considering the in vivo conditions. For biorelevant dissolution, the media is made inline to the in vivo conditions or in vivo gastric media and the intestinal media. Nowadays, ready to use biorelevant medias are available, and these are used for biorelevant media and biorelevant dissolution. Then number 6 question is, what is RSD limit for dissolution? So for initial time points it is not more than 20, and for later time points it is not more than 10. What is dissolution specification? So dissolution specification is the acceptance criteria for dissolution testing. And for example, for IR formulation, it will be like, for example, not less than 80% Q or 85% in 15 minutes or 30 minutes. So that is the acceptance criteria. Then is, what are the medias used in dissolution testing? The medias like 0.1 normal HCl, pH 4.5 acetate buffer, pH 6.8 phosphate buffer, pH 7.5 phosphate buffer can be used. Water can also be used as a media. These medias may be with surfactant or may be without surfactants. Then what is multimedia dissolution? So multimedia dissolution is the dissolution testing where more than one media are used to test the formulation. Like the dissolution parameters are kept same and media are different, like 0.1 normal HCl, pH 4.8 acetate buffer and pH 6.8 phosphate buffer. The multimedia dissolution is done to understand the difference between the media or difference between the behavior of the formulation in different pH medias. Then number 10 question, what can be the dissolution media and parameter if monograph is not available? So if monograph is not available, then in-house method is required to be developed, and the in-house method is developed considering the drug molecule properties, drug solubility, BCS class, and the formulation design. Can different product have same dissolution media? So yes, different product may have same dissolution media, but the dissolution testing is a product specific dissolution testing, and 0.1 normal HCl can be used for many of the products, but the parameters, the specifications will vary. Then what is BCS class? BCS class is the biopharmaceutical classification system or biopharmaceutics classification system which classifies the drug molecules based on their solubility and permeability. So these have 4 classes: class 1, 2, 3 and 4. Class 1 are highly soluble and highly permeable molecules. Class 2 are low soluble, highly permeable molecules. Class 3 are high soluble, low permeable molecules. And class 4 are low soluble and low permeable molecules. 13th number question is, how BCS class is related to dissolution? So as solubility of the molecule affects the dissolution, the BCS class 1 and 3 molecules will dissolve rapidly and may have rapid or very rapid dissolution properties, and rapid and very rapid dissolution will be there for IR formulations. What is rapid and very rapid release? So generally rapid release is the more than 85% release in 30 minutes for the IR formulation, and if the release is more than 85% in 15 minutes, then it will call as a very rapid release. 15 number question is, what are the time points for dissolution? Generally the profiling is done starting from 5 minutes, 10 minutes, 15, 30, 45 and 60 minutes for IR formulations, but the specification time points are generally 15 minutes, 30 minutes, 45 minutes and the 60 minutes. 60 number question is, how dissolution tests differ for IR, ER and DR formulations? So generally for IR formulations, dissolution is run for 1 hour. For extended release formulation, depending on the formulation characteristics, can be run for 8 hours, 10 hours, 12 hours, 18 hours, 24 hours, and may vary based on the product. For DR formulation, acid resistance is testing is done in zero point 0.1 normal HCl for 1 hour or for 2 hours, and after that, the as per the method A and method B, the dissolution is run for 1 hour. So these are the generalized differences between IR, ER and DR formulation dissolution. How the dissolution media is selected. So dissolution media is selected based on the API solubility, formulation characteristics, API stability in the media, and the sink conditions. Then what is sink condition? So sink condition is the requirement for dissolution testing, and if the media and media volume is suitable or can dissolve 3 times of the highest strength of the formulation, or the 3 times of the strength of the formulation being tested, or quantity of the API which is contained in the highest strength, then you can say that the sink condition is met. Sink condition is important because if sink condition is not meeting, then it may happen that because of the solubility problem, the dissolution cannot be reflected as a true formulation behavior. What is done if sink condition is not achieved? So generally surfactants are used. Sometimes the media volume is increased. Different surfactants are tried for getting the sink condition. And if still the sink condition is not achievable, that time the dissolution can be run for is a condition of 1.5x or 1x to the solubility of the higher strength. For example, if you are working on a 300 mg tablet or 300 mg label claim capsule, and media can dissolve only 400 mg or 450 mg of the drug, so also you can run the dissolution, and you have to prove that because of the sink condition, the formulation variation is not being not being affected, and you have to justify that the though the sink condition is not achieved, but still the dissolution testing is having a good discriminatory power. Then 20 number question is, what is S1, S2, S3 level? So these are the acceptance criteria given in the USP for IR formulations, and L1, L2, L3 levels are given for the extended release formulations. Similarly, A1, A2, A3, B1, B2, B3 criteria is given for delayed release type of formulations. Then how to detect sink condition? So sink condition is calculated based on the solubility and the media volume of the dissolution. Solubility of the API is considered, and if the media is sufficient to dissolve 3 times of the dose, then you can say that sink is achieved. What are the surfactants in dissolution? Generally, sodium lauryl sulfate (SLS) and polysorbates are used as surfactants. Whenever the formulation contains low soluble drug substances, that time to achieve the sink condition, surfactants are used. What is Q and Q point? So Q is nothing but the quantity of the label claim that is released or dissolved at some specific time point. That is also called as Q point. For example, the Q may be 80%, and in S1 criteria it will be Q plus 5, that is 85%. But Q is the quantity of the API that is released at specific time point. What problem you have faced for dissolution testing? This is important question, and it is experience dependent question. So whatever the problem you have faced, that you can elaborate. Sometimes the question may be asked that, have you worked on the combination formulation, and have you developed a method for combination formulation? Then what is F2 factor? So F1 F2 factor is the similarity factor and difference factor as per the regulatory guidelines, which is calculated to understand the similarity and differences between the two dissolution profiles, that may be between the reference product and the test product, that may be different, that may be between the two different batches. Is dissolution similarity required for B strength? So generally this question is asked in the interviews, that if you are going for a pivotal biostudy or if you are going for a pilot biostudy and you don't have F2 matching profile, then will you proceed for biostudies or not? So it is dependent on directly onto the formulation. Sometimes F2 can be achieved, sometimes it cannot be achieved. And generally, whenever the biostudies are performed, that time similarity for the dissolution is not mandatory. What is importance of dissolution test? So dissolution test is a performance test for formulation which predicts the in vivo behavior. SUPAC changes can be justified by dissolution testing. Biowaivers can be taken based on the dissolution justifications. Then site changes, manufacturing changes, API changes can be justified based on the dissolution test results. What are the formulation and process factors affecting the dissolution? So based on the formulation type and process type, you can give the examples. Like for IR tablet, concentration of binder, API particle size, disintegrant level, lubricant level, hardness level, granulation time can have impact onto the dissolution. Then the question is how to set Q point. So Q point is set based on the dissolution profile, and generally Q point is set at a time point where you are getting more than 85% of the drug release. What are the dissolution apparatus types? So USP1 is basket, USP2 is paddle, and there are total 7 number of apparatus. Like USP3 is reciprocating cylinder, and USP4 is flow through cell. So always try to remember these all 7 apparatus. Generally the USP1 and USP2 apparatus are widely used. What are the standard dissolution conditions? So generally for USP1 basket apparatus, 100 RPM and the media volume of 500 ml or 900 ml is standard condition. While for USP2 paddle apparatus, media volume of 500 ml or 900 ml and the paddle speed of 50 RPM is standard condition. What is the problem of using water as a media? Generally water don't have the don't used as a media because there is no buffer capacity and water can vary day to day and from lab to lab. Then the question within question is when water can be used as a media. So if the drug molecule is having good solubility at across all the pH range, and if there is no impact of pH of the media onto the drug solubility and dissolution, then water can be used. When alcohol dose dumping studies are used, so generally alcohol dose dumping studies are used for extended release formulations. When 0.1 normal HCl is used as a media, so whenever you are working onto the formulation containing BCS class 1 and 3 APIs and you are getting rapid or very rapid release for IR formulations, generally 0.1 normal HCl is used as a media. How the delayed release formulations are tested? So delayed release formulations are tested for acid resistance test in 0.1 normal HCl, and after that the media pH is changed or sometime the media is replaced, and then the Q point testing is done. When dissolution method development is required? So the question is when dissolution method development is required, and the answer will be Dissolution method is required for all the products and all the types. Whenever you are working on a new or generic formulation, method development is must. And in-house dissolution parameters and method is given when there is no monograph available. But always remember, if monograph is available, still you have to prove the discriminatory power and you have to submit the development report. When peak vessels or apex vessels are used, generally peak vessels are used when there is a coning effect or heat formation for the formulations. And due to that, if you are not getting good release, then apex vessels can be used. But generally, peak vessels are avoided. How to select speed or RPM of USB2 paddle apparatus? So speed or RPM is selected based on the standard conditions like 50 RPM, and if you are not getting the extent of release, then you can increase the speed based on the physical observations like hip formation or cone formation. So from 50 you can go to 60 and then you can increase up to 75 RPM. How to select dissolution media volume? So media volume is selected based on the standard conditions like 500 ml and 900 ml, and also the sink is considered. If sink is not achieved, that time media volume can be increased, and if it is not still possible, then surfactants are used. When sinkers are used, so whenever the formulations float in the vessel or stick to the vessel walls, or if there is ununiform landing of the formulation in the vessel, that time sinkers are used. Sinkers use can give you good reproducibility in the dissolution testing. What to observe during dissolution testing? Whenever you are putting the tablet inside the dissolution vessel and you are starting the dissolution test, that time you should observe how the tablets are landing inside the vessel. Is there any sticking to the vessel wall or to the paddle? And you should observe the physical behavior, whether the tablets are moving inside the vessel, whether these are dancing, spinning or revolving or rotating with the paddle. And you should also observe the disintegration pattern. When USB 3 and USB 4 apparatus is used, generally USB 3 and 4 apparatus are used for non-disintegrating type of formulations and also for the formulations containing low soluble or poorly soluble APIs. What is DPM for USB 3? So DPM is dips per minute, like RPM for paddle. In the USB 3 apparatus, dips per minute is used. And you can develop a method from 5 dpm, 10 dpm, 15 dpm, 20 dpm. Like this, you can perform the dissolution testing and set the dips per minute. Why variations are observed in the dissolution results? So there are lot of reasons for variations. That may be from the formulation point of view, that may be from the dissolution apparatus point of view, or from the analytical point of view. So there are various reasons. Why filters are used in dissolution testing? See, the formulation contains the soluble and insoluble materials, and if you don't use the filters, then the insoluble materials will come out into the sample. If API is insoluble, then if you remove the sample without filter, you will get the insoluble particles, and these particles will get slowly solubilized, and you will observe the variations, and true dissolution behavior cannot be understood. So filters are used to filter the insoluble materials. What to do if drug is not stable in dissolution media? So generally this question is asked for the experience level people, and the answer may be select the media or media pH where the drug is having good stability, and stability is expected till the dissolution run is completed and analysis is completed. Also for some of the molecules you can add stabilizer in the dissolution. What are the frequent regulatory queries for dissolution testing? The frequent regulatory query for dissolution testing is for tightening the dissolution specifications. if you are giving wide dissolution specification, and if the stability data and the biobatch results are not in line to those specifications, then there will be a query or suggestion for tightening the dissolution specification. Also, justification is required for the surfactant level selection, media volume selection, and RPM selection. So these are the general regulatory queries. The last 50 number question is when the dissolution testing parameters and specifications are finalized. Generally, the dissolution specifications and parameters are finalized after the regulatory query is closed and you have got the regulatory approval for commercialization. After that, the parameters are finalized. So these are the frequently asked questions. Dissolution testing and dissolution specification is a very important testing for oral formulations. It is a very vast topic, and you can go through the videos which are there available on this channel, and you can have the good understanding. So whenever you are going for interview, always have good understanding about the dissolution testing. The dissolution and dissolution related questions are always asked in the interviews for the departments like R&D, quality control, quality assurance, production, regulatory affairs, and also for the clinicals. The topic of dissolution is not limited to the department in the pharma, but it is applicable to the all pharma professionals. I have covered some important questions. You can also make a list of your own questions and you can practice the answers. Whenever you get the good understanding, you can write it down or you can save somewhere. So thank you for watching the video. Please do like, share and subscribe to Pharma Learning in Depth channel. Thank you.